We are about to start with Francesco Tadini-Buoninsegni from University of Florence. So I'm particularly excited to have you here, Francesco. He worked in his early career together with Klaus Fendler and Ernst Bamberg, the founding fathers and pioneers of the technology. So today he will talk about investigating charge transport by P-type ATPases with the SURFE technology. Welcome, Francesco.
Thank you. Thank you very much for the kind invitation, and it's my pleasure to be here and to present our studies on P-type ATPases. So we are using the electrophysiological method based on the solid support membrane, or SSM, to study the transport activity of P-type ATPases. So P-type ATPases constitute a superfamily of membrane transporters that are selective for selective transport ions or other substrates, including phospholipids, across biological membranes using the energy provided by ATP hydrolysis. P-type ATPases are present in all forms of life, and they are generally divided into 5 subfamilies, which are specific to different substrates. And these enzymes play very important physiological roles. In fact, for example, they generate essential electrochemical gradients across the biological membranes. They maintain intracellular ion concentrations, regulate cell volume. They also remove toxic heavy metal ions from the cytoplasm, and they're also responsible for the asymmetric distribution of phospholipids across the membrane bilayer. And given their prominent role in many physiological processes, P-type ATPase dysfunction is related to various diseases, including cardiac, neurological, metabolic, inflammatory diseases, and thus P-type ATPases are important targets for a variety of drugs. And these enzymes share similar molecular architecture, which consists of 3 cytoplasmic domains, the A, P, and N domains, and a membrane domain with 8 to 10 transmembrane alpha helices. And moreover, in several P-type ATPases, the N or C terminal extension on the cytoplasmic side behave like regulatory domains. So we have extensively used SSM-based electrophysiology to study the ion transport mechanism of several P-type ATPases, such as the sodium-potassium ATPase, the sarcoplasmic reticulum calcium ATPases, and also human and bacterial copper ATPases. Moreover, we have used the SSM method to study the electrogenicity of phospholipid transport by the mammalian lipase ATP8A2, which translocates the negatively charged phosphatidylserine across the membrane bilayer. But today I will focus on the sarcoplasmic reticulum calcium ATPase. And calcium transporters are found in the plasma membrane and in membranes of organelles, such as the sarcoplasmic reticulum, which is the main intracellular calcium storage organelle. And the sarcoplasmic reticulum calcium ATPase, or SR calcium ATPase, commonly indicated as SERCA, is encoded by 3 genes. So we have 3 main isoforms with different tissue distribution, and in particular, SERCA1A is the predominant isoform in the fast-twitch skeletal muscle, while SERCA2A is mainly found in cardiac muscle. So the SR calcium ATPase couples the hydrolysis of 1 molecule of ATP to the transport of 2 calcium ions against the electrochemical potential gradient from the cytoplasm to the lumen of the sarcoplasmic reticulum. And these enzymes plays a very important physiological role. In fact, by pumping back calcium ions back into the SR lumen, SERCA induces muscle cell relaxation, contributes to intracellular calcium homeostasis. SERCA couples ion transport and ATP hydrolysis in a cyclic sequence of partial reactions which constitute the catalytic cycle. And this slide shows a simplified diagram of the SERCA catalytic cycle. And according to the Albers-Post or E1-E2 scheme, which is the generally accepted model of the catalytic cycle of P-type ATPases, the protein can assume two main conformations denoted E1 and E2 with different affinity for the transported ions and accessibility of the ion binding sites to the cytoplasmic and luminal side. So starting at E1 conformation, the SERCA cycle begins with high affinity binding of two calcium ions from the cytoplasmic side. Then a transient phosphorylated intermediate E1P is formed by the interaction of ATP with a conserved aspartate residue in the cytoplasmic P domain, which is a specific feature of P-type ATPases. Then the conformational transition from the E1P state to the E2P intermediate favors the translocation of calcium ions across the membrane, their release to the luminal side in exchange for two luminal protons. Dephosphorylation of the E2P intermediate is followed by proton translocation release to the cytoplasmic side, thus accelerating the E2 to E1 conformational transition, which completes the catalytic cycle. And in the SERCA catalytic cycle, some reaction steps are electrogenic. This means reaction steps associated with a net charge movement across the membrane. And in particular, the two main electrogenic steps of the SERCA catalytic cycle are cytoplasmic calcium binding and translocation release of calcium ions to the luminal side. The first high-resolution crystal structure of SERCA with bound calcium was solved and published in 2000. Then several crystal structures of the SERCA enzyme in different conformational states in the catalytic cycle were determined at the atomic resolution as shown in this slide. So now let me very briefly introduce the solid support membrane, or SSM, which consists of a alkane-thiol, octadecanthiol monolayer, and phospholipid bilayer supported by a gold electrode. And in particular, the SSM is formed by an octadecanthiol monolayer, which is covalently bound to the gold surface via the sulfur atom, and a phospholipid monolayer on top of the gold-supported thiol layer. And this bilayer, such a bilayer, is characterized by a high mechanical stability so that solutions can be rapidly exchanged at the SSM surface. So once the SSM is formed, vesicles or membrane fragments containing the ATPase of interest can be physically adsorbed to the SSM surface. And then ATPases can be activated by the rapid solution exchange technique. So just would like to mention that the rapid solution exchange SSM method was developed by Klaus Fendler and his co-workers at Max Planck Institute of Biophysics in Frankfurt. And I had the privilege and opportunity to work with Klaus and Jürgen Pinchovius when the technique was developed. And so, very briefly, this slide shows vesicles containing calcium ATPase adsorbed to the SSM surface. In the presence of calcium ions, we perform an ATP concentration jump. The proteins are activated and pump calcium ions into the vesicle. Since the SSM behaves like an electrical capacitor, the positive charge that accumulates on the solution side of the SSM is compensated by an equal and opposite charge on the metal side of the SSM. This means a flow of electrons along the external circuit toward the gold electrode. And this flow of electrons corresponds to the measured capacitive current, which is strictly correlated with the SERCA-generated current and is recorded as a transient current. From the experimental point of view, we use two different solutions in a concentration jump experiment: the non-activating solution and the activating solution, which contains the activating substrate, for example, ATP. So the solution exchange protocol consists of three steps. First, the non-activating solution is conducted through the cuvette, which contains the SSM with adsorbed vesicles containing the ATPase. Then the activating solution is injected into the cuvette and reaction starts. And reaction ends when the activating solution is finally replaced by the non-activating solution. So in other words, exchange from non-activating to activating solution generates the substrate concentration jump at the SSM surface that induces charge movement across the membrane, resulting in a current, in a transient current, which is detected in the measuring circuit. In our SSM studies of the Ca2 ATPase, we have used different membrane preparations containing the SERCA enzyme, and in particular, native sarcoplasmic reticulum vesicles obtained from the rabbit skeletal muscle. And these vesicles are a very useful, very convenient experimental system for structural and functional studies of the enzyme, and contain a high amount of the SERCA1A isoform, which accounts for about 50% of the total protein. We have used also microsomes derived from COS1 cells containing the recombinant enzyme, and proteoliposomes in which the purified Ca2 ATPase was reconstituted. So typical, let's say, current transients generated by the Ca2 ATPase following substrate concentration jumps, as shown in this slide. In particular, if we perform calcium jump in the absence of ATP, we observe a current signal, the red line, which is totally suppressed by the potent and highly selective SERCA inhibitor tapsigargin, TG. So the inhibition experiment confirms that the measured current is actually related to SERCA activity. And in particular, the observed current signal is associated with an electrogenic event in the SERCA catalytic cycle corresponding to binding of calcium ions to the cytoplasmic site. Then, if we perform an ATP jump in the presence of calcium, we observe the current signal, the blue line, which is again totally suppressed by the inhibitor tapsigargin, TG. And in this case, the measured current signal was attributed to a further electrogenic step, let's say, in a SERCA catalytic cycle corresponding to translocation release of calcium ions to the luminal side. So we have used SSM-based electrophysiology to study the ion transport mechanism of the Ca2 ATPase, but also to characterize the effects of pharmacologically relevant compounds. And we found that such compounds affect calcium transport by SERCA and behave like inhibitors or activators of the enzyme. Several SERCA inhibitors of natural or synthetic origin with a variety of chemical structures are known. And these compounds are a very useful tool in structural and functional studies of the Ca2 ATPase, and some of them are also considered as potential drugs. So using the SSM method, we investigated the inhibitory interaction of compounds such as tapsigargin, cyclopyrazonic acid, the antimycotic drug clotrimazole, the phenolic compound curcumin, and also some metal-based anti-cancer drugs, in particular cisplatin. So as you know, cisplatin is a widely used anti-tumor agent for the treatment of several types of tumor. However, cisplatin administration is associated with severe side effects, including nephrotoxicity, neurotoxicity, ototoxicity, and P-type ATPases have been proposed as potential cisplatin targets, usually in connection with the adverse effect of the drug. For example, the cisplatin inhibition of the sodium-potassium ATPase transport activity has been related to the nephrotoxic effect of the drug. And therefore, we are interested in investigating the cisplatin effect on the pumping activity of the Ca2 ATPase. We therefore perform ATP concentration jumps on native SR vesicles in the absence of cisplatin, the red line, and in the presence of 5 micromolar cisplatin. And we found that 5 micromolar cisplatin almost completely suppresses the ATP-induced current signal, thereby indicating that cisplatin strongly interferes with ATP-dependent calcium translocation by the sarcoplasmic ATPase. We then evaluated the concentration dependence of the cisplatin inhibitory effect. And so ATP jumps were performed in the presence of cisplatin at increasing concentration. And this figure shows the dependence of the translocated charge as a function of cisplatin concentration. From the fitting, we determined half maximal inhibitory concentration of 1.3 micromolar. So these results indicate that cisplatin behaves like a potent inhibitor of this sarcoplasmic ATPase. Moreover, we found that cisplatin inhibition of SERCA transport activity is irreversible. And this information may be relevant to the molecular mechanisms underlying the severe side effects that limit the clinical applications of cisplatin and other platinum-based anti-tumor drugs. More recently, we investigated the interaction of SERCA with compounds that are able to stimulate the transport activity of the enzyme. A few SERCA activators are known. For example, the regulatory peptide DORF, the cardiac drug istaroxime, some natural compounds, including, for example, gingerol, and also a few small molecule activators. It has been proposed that pharmacological stimulation of the sarcoplasmic ATPase activity may represent a promising therapeutic strategy to treat diseases associated with SERCA dysfunction. The compound CDN1163 is an interesting example of small molecule SERCA activator. This compound was selected by a high throughput screening of a large chemical library, and it was shown that CDN1163 binds to the sarcoplasmic ATPase and increases the ATPase activity. Well, it is worth mentioning that the compound CDN1163 was tested in a selectivity screen to evaluate its effect on various ion pumps and channels, as was found to be only active towards SERCA. And therefore, we employed the SSM method to study the stimulatory effect of CDN1163 on ATP-dependent calcium translocation by the sarcoplasmic ATPase. And we perform ATP jumps on ATS vesicles in the absence of CDN1163, the initial control black line, and in the presence of 10 micromolar CDN1163 red line. As you can see, we observed a 30% increase in charge movement in the presence of CDN1163. And therefore, these results indicate that the compound has a remarkable stimulatory effect on SERCA transport activity by enhancing ATP-dependent calcium translocation. Then we repeated the ATP jump in the absence of CDN1163, the final control green line, and we observed that the ATP-induced current signal was almost completely restored to the level obtained initially, thus suggesting that the interaction of CDN1163 with the sarcoplasmic ATPase is reversible. We also evaluated the concentration dependence of the CDN1163 stimulatory effect, and therefore we measured current signals generated by SERCA following ATP concentration jumps in the presence of CDN1163 at increasing concentrations, and we therefore determined a half maximal effective concentration site of 6 micromolar. These results shows that the compound behaves like a relatively effective activator of the SERCA enzyme. Now, in our ATP concentration jump experiments, we normally use a high saturating calcium concentration, 10 micromolar free calcium, that enables full activation and maximal current response of the SERCA enzyme. However, it is known that the cytoplasmic calcium concentration in the resting cell is very low, about 0.1 micromolar. And therefore, we examined whether the CDN1163 stimulatory effect could still be observed at low calcium concentrations. And therefore, we performed ATP concentration jumps in the presence of 0.35 micromolar free calcium, and again, in the absence and in the presence of CDN11 or 10 micromolar CDN1163. And we found that at 0.35 micromolar free calcium, so at a low calcium concentration, we observe a 70% increase in charge displacement in the presence of CDN1163. And this result supports the concept that the compound is able to stimulate the SERCA transport activity under physiological calcium concentration. And therefore, we speculate that the interaction of CDN1163 with the calcium ATPase can promote a protein conformational state that favors the release of calcium ion into the SR lumen, thereby enhancing calcium translocation. And finally, I would like to thank my colleagues at the University of Florence, and also Fabio Arnesano, Giovanni Natile, University of Bari, Italy, and Howard Young, University of Alberta, Canada. And thank you very much for your attention.
Thank you, Francesco. Are there questions from the audience?
Many thanks for your talk, very nice talk.
Thank you.
And I see also some name of my colleague in my department, so that's I'm very glad for this collaboration, long-lasting collaboration.
Yes.
And one question is related with the activators, just a curiosity. Do you know some other activators that, because you just at least some class, but you know some other activators that, I don't know, are in clinical trials or something that works in this on SERCA?
Yes, well, actually, we also investigating the effect of a natural compound, gingerol, on SERCA transport activity, and we found that also gingerol is able to stimulate the SERCA transport activity. There is also another compound, actually, in clinical trial application with the cardiac drug istaroxime, which is believed to enhance SERCA transport activity, actually the SERCA2A, so the cardiac isoform of the SERCA enzyme. And we also did some measurements to study the effect of istaroxime on the SERCA2A isoform, and we got results that supports, this activating effect, stimulatory effect on the enzyme.
Okay.
And but generally, I mean, there are also other small molecule activators that are under investigation, and some recent papers reported on small molecule SERCA activators rather effective towards the enzyme. So
Okay.
there are a few SERCA activators known. Yes, it's difficult to find an activator of the enzyme. It's rather, I mean, it's generally more easy to find an inhibitor, but a few examples.
Do you use SR vesicles?
Yes, usually SR vesicles, but I also tested these compounds on protoliposomes, in which the SERCA, purified SERCA enzyme was reconstituted. Also in the case of CDN1163, I check, I mean, I did measurement both enzyme preparations, and they were consistent. I mean, I could get some nice results also with the protoliposomes containing the purified enzyme. This was important because in the SR calcium ATPase SERCA that actually is regulated by a transmembrane protein, sarcolipin, which is, so we wanted to see whether the effect of CDN1163 was a direct interaction on the enzyme, or whether an indirect interaction by affecting sarcolipin action, let's say. So that's why we purified the protein, reconstituting protolipins were containing only the purified enzyme without sarcolipin, and we could reproduce these results. So we can say that the CDN1163 acts directly on the SERCA enzyme. And I think, given the lipophilicity of the compound, we propose that the binding site is actually located in the transmembrane domain of the enzyme. And in fact, some very recent cryo-EM structure of the SERCA enzyme with the CDN1163 identified a binding site in the transmembrane domain. This is not published yet, but very soon. Yes.
Okay, thanks.
Oh, I'm just wondering a very general point. Are you in any way limited by the speed of the concentration change that concentration jump? How fast is it? And
Well,
would it be of any consequence if you could make it much faster?
Well, you mean the time resolution?
Yeah.
Well, in our setup, the time resolution should be around 20 milliseconds. But I'm using a gold sensor, 3 millimeter diameter. So you can use sensor with a smaller diameter, 1 millimeter, and increase the time resolution of the measurement.
But the time resolution will also depend on the speed of your concentration jump.
Right. So we are using a high saturating ATP concentration.
Mm-hmm.
So, I mean, this is not a problem when you use a very high oversaturating substrate concentration, like in the ATP concentration jumps I just showed.
So you're not limited by that?
I'm not limited by that. In this particular case, I'm not. I'm using particular experimental conditions.
Thank you very much for the very nice talk.
Thank you.
Very interesting, the activatory effect of the CDN11. Is it known whether this compound interferes with the beta-adrenergic stimulation of the SERCA? Because under physiological conditions, at higher adrenergic stimulation, the SERCA activity is increased, right?
Mm-hmm.
So is the compound then still active and it can pump more, or is it known? I'm just curious, sorry.
To be honest, I don't know exactly. And so has been used in certain cell lines, animal models, and but to be honest, I don't know exactly if these studies have been done. I know that it's in preclinical studies for diseases like neurodegenerative diseases, metabolic disorders. They are using animal models of the type 2 diabetes and see the action of the compound, these particular models.
All right, then thank you, Francesco.
Thank you very much.